We'll get started with our block visiting professor rounds. I'm honored to introduce Dr. Michael Wodlansky, who's from the Medical College of Wisconsin. He's a professor of medicine and pharmacology. He's also the director of research of cardiovascular medicine, and he's also a co-director of the cardiovascular translational research unit. Mike did his undergraduate work at Stanford. He did his medical school. He returned to the Midwest and went to the University of Michigan. He did his residency in medicine at the Brigham and Women's Hospital and stayed in Boston to do his cardiology fellowship at Boston University. He did clinical research training with Dr. Joe Vita. He did work in this group in terms of measuring endothelial function in patients, and was in one of the preeminent clinical vascular biology groups. He's done a lot of work in terms of measuring endothelial function in metabolic syndrome, diabetes, the impact of several supplements on endothelial function, and he's made major contributions in this area. The one nice thing about Mike's research work is most of his research is done in humans. He's developed a lot of techniques to measure endothelial function in adipose tissue. He has been in his academic career mainly at the Medical College of Wisconsin. He was Associate Cardiology Fellowship Director for many years, and between 2014 and 2019, he was the Fellowship Director. He's mentored over 20 postdocs and cardiology fellows, and many of them are still in academic positions. So I'm very honored to introduce Dr. Wodlansky, and we look forward to listening to his presentation. Mike?
Thank you, Francis. So greetings from the home office in Fox Point, Wisconsin. We're about 5 minutes from the shores of Lake Michigan here. Obviously, it would be nice to have actually come out and visit, but we can certainly go through what information I have to share this way. Let me get started here. These are sort of, let me go ahead and see if this will advance. Hold on a second. All right. All right, these are my disclosures here. The grant that's most relevant to some of the work that's actually being shown here is the one in italics there from NHLBI. Of late, we've actually gotten some support actually for the Lactobacillus product we've been using from the company called Probiotic. They were not involved at all in the research actually being shown here. We're more interested in the Lactobacillus itself, and no off-label drug use we're going to be discussed today. So there's some quick definitions so we get everyone on the same page. So when we refer to the gut microbiota, it refers to basically the totality of the bacterial, viral, and protozoan composition of the GI tract. When we refer to the microbiome, it actually refers to the genetic composition of the gut microbiota, which is actually about 100 times more rich in genes than the human genome is. When we talk about and refer to prebiotics, those refer to foods or other products that may induce favorable growth or activity interaction of the gut microbiota with potentially to improve health. And when we talk about probiotics, those are actually live microorganisms which may provide favorable health benefits. Postbiotics are actually the products that are made by the gut microbiota that may impact health. So when you ingest a pre or probiotic, what the microbiota actually produces, those metabolites, are what are known as postbiotics. And then not at all about microbiota or postbiotics is endothelial function. Just to define it here before I talk about it in relationship to the science, basically refers in general to the ability of the vascular endothelium, the cells that line blood vessels and the media above the lumen of the vessels, to regulate the activities and maintain vascular health. And there are a variety of different manners and paracrine and autocrine ways in which the vascular endothelium does that. One of the primary molecules that does it is nitric oxide, and to maintain vascular health. Before I get into sort of some of the studies of what we did as far as probiotic supplementation, vascular function, we get into a little bit of history of the field and how we got to where we are today. So this here is actually a guy named Pliny the Elder. Pliny was from a wealthy family. He served in the Roman army, served several Roman emperors. He actually died from smoke inhalations from complications from the eruption of Mount Vesuvius. But his biggest contribution that is known from antiquity was actually the development of what was thought to be one of the first scientific encyclopedias known as natural history, which is seen here on the right side of the screen, which is actually considered to be a tome of scientific information from antiquity and through the Middle Ages. It contained 37 books, 14 of them focused on medicine and drugs. And in here, he actually mentioned the use of fermented milk products to help stomach ailments. And this is perhaps the first mention that we've seen in sort of the ancient literature of that. And while this might seem sort of prescient of him, he also had a variety of different things that didn't sort of pan out. He also thought elephants would only board ships if they were told by the captain they'd be returned home when the trip was completed, and that mythical dog-headed creatures and humans with one huge foot habitated the edges of the world. So not everything in there was completely correct, but this is but he was credited as one of the first persons to actually mention potential use of probiotic products for health. And it wasn't until nearly 1800 years later plus that Elie Menshnikov sort of revived a bit of this field. Dr. Menshnikov was born in the Ukraine. He got his PhD from the University of St. Petersburg. He was actually best known for his work in immunity. He was one of the first persons to recognize that the phagocyte was the first line of defense for acute infections. And he actually won the Nobel Prize for that work in 1908. He's also often referred to as the father of probiotics because he was the individual who actually observed that longer lifespans in individuals who were in the lower classes or the peasant classes of Bulgaria. They often outlived nobility despite harsh conditions. And he attributed this to the consumption of fermented milk products. And he actually surmised that it was actually potentially the lactic acid in these bacteria in their yogurt that had the anti-aging properties. That bacteria, which later became known as Lactobacillus bulgaris, and it was sort of his observation, and he sort of published these in The Prolongation of Life, which you also see here on the right side of the page. About the same time, Henri Tissler at the Pasteur Institute in Paris had actually noticed in the gut and stool of breastfed infants these Y-shaped bacteria, these bifid bacteria, and he reported that infants with these bifidobacteria actually in the digestive tracts had fewer gastrointestinal problems, as evidenced by fewer diarrheal illnesses. In the cardiovascular field, this is microbiota. It's sort of been a sleeping giant for a long period of time. If you actually go to PubMed and you look for microbiota and atherosclerosis, you find very little there, and then suddenly start seeing some building sort of late zeros, and then as we get into the last decade, you start significantly larger number of publications in this area. Now, this wasn't because of lack of sort of thought that potentially that a bacteria may play some role in the development of cardiovascular disease. There were observations, as many of you may know, in the late 1980s of chlamydia pneumoniae in atherosclerotic plaques, and that led to multiple trials actually sort of summarize some of their big main figures summarized here, looking at trying to treat atherosclerosis with antibiotics. The Wizard trial, the ACES trial, and Prove It, or TB22, all published in the early zeros, essentially showed the treatment with azithromycin or gedafloxacin to attempt to eradicate these bacteria did not have any significant impact on cardiovascular outcomes. However, there were some signals still in the early zeros that potentially the microbiome biota might actually have some impact on cardiovascular risk. This is a study that actually was published in the New England Journal now about 14 years ago, 13, 14 years ago, by a group looking at periodontal disease, and there's been a lot of epidemiologic literature sort of linking the association between periodontal disease and atherosclerosis and cardiovascular risk. And so what they did is they took a small group of individuals with periodontal disease and they randomized them to usual care for that disease or to a much more aggressive a regimen of removing teeth that needed to be, the rod needed to be removed, very deep cleaning, and look to see how that affected blood vessel function by flow-mediated dilation, which you see on by the graph on the left side of the screen here on the y-axis, and looked at how that changed in the intensive treatment group versus the control group. And what you actually see in that graph is that early on day one, actually, the intensive treatment group has sort of this plummeting of FMD, flow-mediated dilation of the brachial artery, suggesting some early on impairment, which may relate to some transient bacteremia or more progressive treatment. But then over time, actually, their endothelial function improved. So there were some signals, at least early on, that potentially you could modulate, by modulation or treatment of periodontal disease and changing the oral gut flora, that potentially you could modulate risk. It was really a study actually published now, in the early 2010s by Stan Hazen's group, which looked at the intake of foods high in phosphatidylcholine as it might affect cardiovascular risk. That really kind of helped reignite some of this field. Now, phosphatidylcholine is contained in many common foods people eat: shellfish, meat, eggs, poultry. And when ingested actually into the gut, the phosphatidylcholine is broken down into choline and then subsequently actually converted by the gut microbiota to trimethylamine or TMA. That TMA then is absorbed, reaches the portal circulation, gets to the liver, and is converted by hepatic flavin monoxidase enzymes into a compound known as TMAO or trimethylamine N oxide, which many of you have maybe heard of. There's a lot of effects that have been shown in vitro and animal models looking at its ability to cause vascular inflammation, oxidative stress, and be prothrombotic. And in fact, as part of this work, they published data looking at patients who presented to the Cleveland Clinic's cath lab and measured at the time of presentation their plasma levels of TMAO, then followed them out for 3 years looking for risk of myocardial infarction, stroke, or death. And what they found is individuals with the highest quartiles of circulating TMAO had the highest risk of adverse cardiovascular events over that 3-year period in a subject group of around 4,000 patients. So that helped ignite the field, but a lot of other areas of interest sort of popped up too as well. So people were looking at whether or not metabolites that are produced by the microbiome might actually be affecting vascular health, looking at microRNAs that might be influenced as far as their production in the vasculature related to those metabolites, and the use of prebiotics. So we could go through the field for a very long time, but also to summarize what we kind of know was the background work that's been found. So current evidence actually does support gut vascular access related to regulation of vascular health that may relate to a variety of different factors, including TMAO, production of short-chain fatty acids from the gut microbiota, bile acids, or gut permeability. There's some limitations we need to realize some of the evidence that we had from this, which is that translation from animal models to human models is a little bit challenging, particularly in this field. And the gut microbiota in animals is significantly different than you see in humans. Not only that, the common sort of models that we see for atherosclerosis, particularly murine models, their immune systems are significantly different than that of humans, so it becomes difficult to translate a lot of the interventional studies in animals to humans. On top of that, a lot of the human data was cross-sectional or epidemiologic, making it difficult to sort of to translate those findings to actual causality. So that's where the field sort of stood. So the question really becomes like, what kind of things can we target the gut microbiome, and to those kind of interventions to potentially impact human vascular health. So this is kind of where my story began. I'm going to sort of run through the data and get to the work that we did based on sort of how I ended up actually in doing this work in this field. So back now, published about 8 years ago, John Baker, who's a professor in the Department of Cardiovascular Surgery at Medical College of Wisconsin, and Nina Saltzman, who heads our Center of Microbiome Research, did some work actually in dull salt sensitive rats, which are rats that get hypertension when they're exposed to high salt diets. And what they were looked at actually was the influence of vancomycin as an antibiotic, as well as Lactobacillus plantarum 299v on infarct size in these rats. And so what they actually did is they fed them either in their water, they actually included vancomycin, or they fed them the Lactobacillus and the plantarum in the form of a fermented oat product. And they did this for several weeks, and then they actually induced an infarct by ligating the left anterior descending artery for 30 minutes, reopening, and then closing again after 2 hours. And they looked at infarct sizes, and they looked at recovery of left ventricular development pressure in Langerhans models. And what they found is in the animals which they gave good belly to, which is the product that actually contains the Lactobacillus plantarum 299v, that they had a significant reduction in the size of the infarcts. That if they actually gave them leptin, which is a adipokine, which is made by adipose, which is known to drive inflammation, particularly through the IL-6 pathway, they were actually able to obviate that favorable effect on infarct size. And it also improved, the product actually improved LV developed pressure post-infarct in those animals. Vancomycin actually showed a similar finding as well. Just as an aside, so this is the product they actually use, is Good Belly straight shot product that actually, it's a fermented oat product that has 20 billion colony forming units of Lactobacillus plantarum in and of itself. So what was known around the time in humans, and part of what drove them to take a look at Lactobacillus plantarum 299v, was some studies in higher risk individuals, humans who were either smokers or had elevated risk, and they looked at supplementation for several weeks with Lactobacillus plantarum 299v, and looked at inflammatory markers IL-6, which seemed to go down. And then you took monocytes from those individuals and ran them over a plate which had cultured human umbilical vein endothelial cells on them to look at adhesion, and found that actually they reduced adhesion of those cells, those monocytes, from individuals who had been supplemented with Lactobacillus plantarum. That was really kind of the extent of the knowledge base at that time. And so then they came to me and actually asked me, how would we translate their findings of reduced infarct size to humans? Now, we had no money, and I knew doing a study in individuals post-infarct and doing a lot of cardiovascular imaging, echocardiography or cardiac MRI would be difficult and expensive to do. And looking at their data, I thought, well, maybe what some of the effect here was an improvement in perfusion and potentially vascular function, and maybe that was part of the contribution we're seeing. So maybe this was a vascular effect. And so we proposed looking at Lactobacillus plantarum supplementation in individuals with coronary disease, and would be looking for some mediator that might actually improve endothelial function by increasing nitric oxide production from the vascular endothelium. We actually put in a small pilot grant that got funded, and so we were off and running. So what did we do? So again, still a small amount of money, and really not enough to really run a large sort of randomized definitive clinical trial. So we ran basically a small interventional study. We recruited 21 men with known coronary disease, and we looked at men only in the small pilot, in large part because of monetary limitations, and all the rats that they had studied were male. I will say that the current clinical trial that we're doing is actually powered to look at both men and women to be able to see if there are differential effects, which may be the case since the microbiota of women and men do differ. So in that particular, but in the pilot study here, we took men with known coronary disease, ages 40 to 75. We excluded any individuals with LV dysfunction, uncontrolled hypertension, anyone who'd been on antibiotics for the last 12 weeks, and anyone with a recent statin change. And we basically gave them 20 billion colony forming units of Lactobacillus plantarum 299v daily for 6 weeks. They then had a 30-day washout period, and we also had the optional vancomycin arm because of the animal study. We made that optional in large part because we weren't sure how many individuals would be up for the potential GI side effects of taking oral vancomycin for 10 days. So we didn't want to dissuade people enrolling who may be interested in doing the first arm of the study, but not the second arm of the study. So our primary outcome, as I've sort of briefly mentioned about before, is brachial flow-mediated dilation. For those of you who are not familiar with it, I'll describe it here in some detail. What you see on the first image on the left side of the screen here is actually my arm, circa 2002, with one of our techs from Boston actually imaging the brachial artery there in longitudinal cross-section, which is on the upper right side of your screen. And then we also use spectral Doppler to measure the flow velocity as well in the vessel. So we then actually take that blood pressure cuff, and actually in our lab we now use the blood pressure cuff below the cubital fossa rather than above the cubital fossa. But regardless, we occlude for 5 minutes. That leads to vasodilatation of the vessels distal to the occlusion. And so when we release after 5 minutes, we get hyperemic flow, as you see on the bottom right side of the screen there. That hyperemic flow, that laminar shear across the vessel, leads to activation of shear responsive elements on the endothelium, ultimately leading to the production of nitric oxide and vasodilatation. And you can represent flow-mediated dilation essentially as an absolute difference between baseline and peak vessel diameter, or as the percentage of increase from baseline. This has been shown now in multiple studies, 20 to 30 studies at least at this point, in populations both with and without coronary disease, to be a marker of increased, marker of risk stratification for cardiovascular events. So what did we see? What about the study? So what did we actually find? So the group of individuals we had here is actually a pretty standard group of individuals we see with coronary artery disease, average age in the early 60s, only a little bit smaller number of diabetics than we may have expected, half of them with hypertension, many of them hyperlipidemia, most of them on statins. Most of them had quit smoking, only a small number had actually still currently smoking, and with a body mass index over 30. This is their medication regimen, pretty standard regimen that we see in a lot of our patients with established CAD. Probably maybe a little bit lower in ACE inhibitor use, although and ARB use, although that may relate to the fact that these were patients specifically chosen who didn't have LV dysfunction. So pre and post probiotic, looking at a lot of the data, there was no really difference in weight, BMI, heart rate, or cholesterol levels that we found. We did find a little bit of an increase in blood pressure. This was the blood pressures that were taken from these individuals in triplicate when they first showed up for their visit. It actually didn't play out. So when we do flow-mediated dilation, we actually have them rest for 10 minutes in a quiet room without any other disturbances to sort of make sure we have a good resting baseline. And we take then, prior to actually doing brachial artery measurements, we actually measure blood pressure again at that point in triplicate. And blood pressure, if you compare pre and post at that time, didn't change. The larger trial, we haven't seen, it doesn't seem to be any signal there either, but we'll see in the larger trial whether that actually plays out. So what were the primary results here? So when we looked at brachial flow-mediated dilatation, so week 1 was their first visit, and then actually 6 weeks after supplementation, we saw an increase in flow-mediated dilatation by about 25% relative increase. It actually dropped back down towards baseline at 30 days post-completion. The vancomycin arm actually didn't, the FMD was no different than baseline, so that didn't change anything. When we looked at leptin levels pre and post, this was found in the animals to be a significant modulator, something modulated by the probiotic. We did see leptin levels did drop post-supplementation. We didn't see any change in the adiponectin levels. We did see drop in inflammatory markers, IL-8 and IL-12, known to be related to the development of atherosclerotic disease. We actually did get stool from all these patients, and we actually sent that for 16S DNA analysis to identify whether or not there were significant changes in the gut microbiome, biota itself with supplementation. The results you see here at basically the class level, there were no real changes in, at the class level of the gut microbiota. And we really didn't see any significant change in clustering based on supplementation here. It's based on the principal component analysis you see on the right side of the screen. When we looked a little bit deeper at the operational taxonomic unit level, which is really kind of the species/genus level of gut microbiota, we did find there were 70 operational taxonomic units that did significantly differ prior to adjustment for multiple testing. Obviously, with a group of 20 individuals after multiple testing, those drop out, but it did sort of give us some hypothesis generating ideas. And of those, 6 of the highest ones were these Lactobacillus species. So what do we know so far? So we know that Lactobacillus 299v supplementation led to an improvement of endothelial-dependent vasodilation. It seemed to reduce leptin levels and reduce circulating IL-8 and IL-12 levels, there was no didn't really see any change at the class level in the stool of these individuals, but potentially some changes at the genus and species level. What we don't know is really, what it might be doing actually to improve human endothelial function or reduce inflammation. And that's sort of what are these mediators of response, and that's sort of where more of our work is sort of concentrated on these days. So when thinking about this, there are a couple of populations of cells that we really need to think about with respect to how this probiotic might actually be affecting vascular health. One would be to look directly at the endothelial cells. The other is to look at the inflammatory cells. There's clearly an anti-inflammatory effect, and as we all know, the inflammation is clearly a significant player in modulating the development of atherosclerosis. So in our lab, we approach this to try to actually get some mechanistic specificity in human tissues. We need different tools to do it than just some of the in vivo measurements. And there are a couple of common ways in which we do this. The first is you see, excuse me, on the left side of the screen, is by obtaining intact human resistance vessels that we can get either from surgical specimens that we get from the OR or from individuals who volunteer in some of our studies to have gluteal fat pad biopsies. In those studies, we actually do about a 1 to 1.5 centimeter incision in the upper outer quadrant of the non-dominant side, non-dominant side of those individuals. If they're right-handed, we do the left side. The left-handed, we do the right side. And we obtain usually about 2 cc's of adipose out of those incisions, and then close those up. And we usually can get at least 2 to 3 vessels, which we can dissect out of those tissue samples, usually approximately about 100 microns in diameter. And then we can take those vessels, actually put them in small microorgan chambers and suspend them in those microorgan chambers and actually test vasodilatation to different stimuli. The other thing we can do actually is intraluminally transfect those vessels with different either small interfering RNAs antimeres or mimics to try to molecularly modulate targets that we're interested in and look at how that affects vasodilatation. What you see on the sort of screen there on the left, in the upper portion of the left side of the picture on the screen, is actual human vessel. Actually, there is a light center there is actually in the lumen there, and then the walls of the vessel you see on the side. That's actually a nice sort of tool that we can use in large part because it's an intact vessel, and we can look at functionality as it might relate to some pathways we're interested in. Another method which we can use actually is to obtain cells off of wires that are used either for placement of venous or arterial lines. We can capture endothelial cells that way. We identify them as being CD31 positive and 45 negative. And then the larger issue with those cells is what we can actually do, look at some protein measurements. We've been getting better as far as the yields are concerned and being able to do things like RNA-seq or even single-cell RNA-seq on them. But the number of cells is small, so there's some limitations to what you can do with them. But these are some of the ways in which we can approach in the lab using human tissue samples and try to approach some of the specificity and molecular techniques that you might see in animal models. So the question really is, going back to some of the research, what are the metabolites and what other things might be affecting that vascular function as it relates to supplementation with Lp299v? And the obvious first question based on prior data and everyone else's work is TMAO actually a player here? Is that what we're seeing? So we did measure TMAO, and we didn't see any significant difference between pre and post supplementation. In fact, if anything, we actually saw an increase. It's interesting, you see on the right side of the screen there is actually the individual subject data. You know, there were these two subjects here that had these significant increases in TMAO pre versus post. I suspect those are a couple of individuals who ate prior to coming and forgot the instructions. But overall, if you even look, you take those two out and look at the rest of them, there was no real significant change in TMAO. We also actually took those vessels I talked about, some from human arterioles, we put them in our microorgan chambers, and we actually exposed them to TMAO at 10 micromoles for about 4 hours. And if you remember way back from the graph I showed you from the Cleveland Clinic data, 10 micromolar is a little bit higher than what we saw in the highest quartile of risk in those in the Cleveland Clinic clinical data. We didn't really see any difference, at least acutely, in dilation of those vessels. They were exposed to TMAO. Now, that doesn't mean long-term there could be effects, but at least as an acute effect, we did not see that in these vessels. So the question is, what else could that be? And one thing, you know, which I brought up briefly earlier, could be is these short-chain fatty acids. Now, basically short-chain fatty acids in humans are overwhelmingly produced by the gut microbiome. By 99% of them in circulation are short-chain fatty acids are created by the gut microbiome. It's in large part because there are a lot of dietary fibers and carbohydrates or fatty acids that we may take in that we don't have enzymes to digest, but the gut microbiota does. And they break this down into these small short-chain fatty acids that can then come across, be absorbed into the bloodstream, and have a variety of different effects. So we looked actually at levels of 3 of the more common ones: acetic acid, propionic acid, and butyric acid, and pre and post supplementation plasma. And we found actually a drop in acetic acid levels and an increase in propionic acid levels, and basically no change in butyric acid levels. So going into sort of how these short-chain fatty acids might be signaling in the vasculature, there are several receptors a couple of receptors that might be active. One is this olfactory 78 receptor, which is located on vascular smooth muscle. The other is GPCR41, or known in humans as FFAR3, which has been found to be located on the vascular endothelium. And actually in animal models, and has been shown that knocking out this receptor actually leads to the development of hypertension in an endothelium-dependent manner. What's also interesting about this receptor is it has a very high affinity for propionic acid, much higher than acetic acid and definitely higher than butyric acid. So the changes we see, we suspect it might potentially could those be related to modulation based on propionic acid levels. And so we basically, again, took the vessels, those microvessels, resistance vessels that we get from human tissues, and we did some studies in those. And we first did is we actually took paired vessels from individuals with coronary artery disease, and we exposed them to either pre-supplementation plasma or post-supplementation plasma for about 6 hours. And we looked at vasodilatation to acetylcholine, which dilates these vessels in endothelium-dependent based manner based on activation of muscarinic receptors. And what we found actually is that if you gave CAD vessels from coronary artery disease patients and you expose it to post-supplementation plasma, these red circles here, you actually see an improvement in endothelium-dependent vasodilatation. And if we use L-NAME to actually block the production of nitric oxide, we actually find that effect is actually completely blunted. It appears to be an NO-related effect. So some factor in the plasma seems to lead to a favorable effect on endothelium-dependent vasodilatation related to NO bioavailability. When we take these vessels actually, and we knock down the FFAR3 receptor using an siRNA protocol, which we designed, we actually find that the vasodilatation to acetylcholine actually goes down significantly. Also, again, I highlight for you here in the middle graph that the propionic acid concentrations increases that we saw was about 5 micromolar, which doesn't seem like much. But if you actually look at the IC50 for this receptor, it's right in the range between 10 and 50 micromoles. So small changes can lead to significant changes in the activation of the receptor. When we actually look at maximal vasodilatation of these vessels, it's either 30 or 35 micromoles of propionic acid. We do actually see a significant increase in vasodilatation. But it's plausible at least part of the effect might actually be due to propionic acid. It's obviously an area we're still working on. So there could be other metabolites though beyond just the short chain fatty acids. So we actually did send the plasma to the Mayo to actually do some analysis, unbiased analyses to look at metabolic profiling, looking at the metabolome. The blue squares here are actually the post-supplementation plasma samples. The red triangles are the pre-supplementation plasma samples. I think what you can kind of appreciate here is you can almost pick out which sample is pre and post based on the metabolomic profiling. When we look at this, there are 25 known compounds and 85 unknown compounds that were significantly changed prior to multiple testing adjustment, giving us some potential directions to look at, and we're still kind of sifting through a lot of that data now. But clearly there may be other factors other than the short chain fatty acids as well that are at play. The other side of the equation that we've been looking at is the inflammatory side of the equation. So, whether or not the effect of supplementation on inflammation directly from mononuclear cells might actually be modulating some of the effects we see. And to do some of this work, we actually collaborated with Marty Hessner, who's the head of our Juvenile Diabetes Center here at Medical College of Wisconsin. And Marty actually helped put together this plasma-induced transcription assay that was been validated by the Juvenile Diabetes Foundation, where basically we take mononuclear cells from a single healthy donor, and we can expose it to 2 separate types of plasma exposures. In this case, we're exposed to pre-supplementation and post-supplementation plasma, and then essentially look using the Illumina chip, essentially to look at transcriptome changes that may be related to exposure to that plasma. So in doing these experiments, this is what we found. So these are the heat maps here. So the first thing we found kind of interesting was that there were nearly 1,400 genes at least a 20% change in expression pre and post supplementation. using a generous FDR here as we were sort of doing more exploratory work. And if you actually, there's an index called the Composite Inflammatory Index, or the II.com, where you can take basically the transcript products for anti-inflammatory versus pro-inflammatory genes, and to look to see, overall whether or not what you're exposing them to, the cells to, is pro or anti-inflammatory. And that was found that actually the plasma, the post-supplementation plasma, was actually significantly anti-inflammatory. The other interesting thing we found actually is, if you'll see here highlighted, so we have a group of 15 of these individuals that are their subject numbers are in black, and then 4 of them here are in red. What we found interesting in this pattern is that individuals who reported to be daily drinkers had a significantly different, their plasma caused a significantly different heat map relative to the individuals who were not daily drinkers. And that is of interest. In fact, daily drinking of alcohol actually can increase gut permeability and may significantly alter some of the findings that we've seen. And so we kind of, as we went further in this type of work, we separated out the alcoholic subjects, or not alcoholics, the daily drinkers, and focused primarily on those who are non-daily drinkers to see the effects. I think studying the effects in those who drink more regularly would be an interesting area for further work. When we went back and broke down, so again, just the 15 individuals were non-daily drinkers, the findings related to brachial flow-mediated dilatation and inflammation actually held. So, those findings actually held. And again, we didn't find any changes in the operational taxonomic units after adjusting for multiple corrections. When we took these data from the 15 non-daily drinkers and we put that into Ingenuity pathway looking for pathway analysis, we did find significant downregulation of interferon signaling and pathways related to innate immunity. And we ran this through David and found that also to sort of validate these findings from pathway, from IPA. Looking at upstream regulators, we found again significant downregulation of signaling from a lot of the innate immunity pathway from toll-like receptors, and particularly interferon-related signaling, and TNF-alpha as well. Then we went a little bit further actually, and we took our data to look for interactions between the gut microbiota composition changes, the changes we saw in transcriptomes, and some of the changes we saw in measurements of endothelial function. We didn't really find any changes in gene expression as it might correlate to changes in operational taxonomic units. We did find a couple of genes that actually appeared to be inversely correlated with brachial flow-mediated dilatation. One of them was GBP1, it's guanylate binding protein 1, which is known to be induced by interferon alpha and can increase expression of this in activated endothelial cells. And the other was TRAIL, or TNF superfamily member 10, which is also expressed by inflammatory cells and can be found in vessel walls. And there's some conflicting data with respect to how it might affect overall atherogenesis. And so some of what we've been working on now, at least in the mechanistic models, is looking to see whether Lp299 supplementation, whether it be through propionic acid and also looking at additional metabolites, potentially might be affecting vascular function through FFAR3 activation, but potentially through a couple other gene activations as well. So this is very early working model. We were very sort of young in this field and have a lot of discoveries to make. But we started to try to draw at least a few lines here to see whether to try to get some insight into how one way in which a probiotic supplementation might actually be affecting vascular function. So I'll wrap up here. Basically showing that we were able to show here, at least the Lp299v supplementation appears to improve endothelial function in individuals or humans with stable coronary disease. That appears to reduce systemic inflammation in these individuals. But the improvements do not appear to be related to TMAO. They may relate to short chain fatty acids, particularly to propionic acids. We have further work to do there. And there's a lot of work to do with respect to potential mechanisms as it relates to specific inflammatory pathways or gene alterations within the vascular endothelium. So as I sort of alluded to earlier, right now we're in the midst of a clinical trial, which is a randomized placebo-controlled crossover study with sex and BMI balance and stratification to look at whether Lp299v improves endothelial function and has these similar effects in both men and women and individuals with BMIs that are both elevated and not elevated. That trial was actually doing well and ongoing. Obviously, at this point, we're a little bit on hold due to the pandemic. Hopefully, we'll be able to get that back on track once we have gotten through the pandemic. We've been trying to identify and test some bioactive molecules in the plasma. That work is ongoing too as well. We're working on some of the gene expression data too as well to look to see how that correlates. We've been actually able to get do RNA-seq on the endothelium from the vessels that we expose to plasma, and that data was collected and being run to be able to see whether or not we can get some more specificity at the endothelial level as far as gene expression changes. With that, I will wrap up. This is my lab here. It takes a village actually to do all of this. I showed the pictures of John and Nita earlier and Marty. I have had 4 MD cardiologists, postdocs run through my lab during the time of all this work, which helped sort of contribute a lot to this. So thank you.